This is part 2 of the chapter 3 lecture notes or lecture recordings. And we're just looking here at the glycocalyx is being shown in this picture. The glycocalyx is all these green hexagons are glucose molecules. And so we're seeing chains of glucose molecules attached to, if they're attached to a protein, we call it a glycoprotein. And if they're attached to a phospholipid, we call it a glycolipid. But they form the glycocalyx of the plasma membrane, which helps with cell recognition and tissue compatibility. Then there's a video animation on membrane structure. We had talked about that there are junctions that connect cells. They're called cell junctions. There are 3 kinds: tight junctions, gap junctions, and we talked about those 2 types, but I didn't mention desmosomes. So tight junctions form a tight seal and they prevent leakage. And then gap junctions have a gap or a channel that runs through them. And we see basically coordinated activity when you have gap junctions that connect cells. So it allows electrical signals in the form of like ions, charged ions that move through the cells very fast. They're electrical signals, and that allows those cells to contract or move together in a coordinated way. Then we have desmosomes, and desmosomes are like a very strong spot weld. They are very, very strong connections at one spot. And what they do is they keep cells from tearing apart. For example, if you have ever had a sunburn, and I realize that some of you have never had a sunburn, your skin may be dark or you may never have gone out in the sun, but if you've ever had a sunburn or seen someone who had a sunburn and their skin, after a few days after they got the sunburn, their skin started peeling. So when the skin is shed, it is shed in sheets. You can peel it off in sheets. Or even if you were to look at your skin that gets shed in the case of like dandruff on your scalp, or just skin flaking off, if you were to look at it under a microscope, you would realize that those flakes of skin are actually many, many skin cells that are attached to one another. So they're attached and they don't pull apart when the skin is shed because of desmosomes. So desmosomes are very strong connections between cells. But just at, strong connections at like one spot. So they almost look like a button. All right, now we're going to talk about membrane transport, and that is transport across the plasma membrane, either into the cell or out of the cell. There's 2 main types. Transport that is called passive transport does not require energy, and substances move from their high to their low concentration. So they move from high to low, where their concentration is high to where it's low. And then active transport does require energy because substances are moving against their concentration gradient. They are moving from where their concentration is low to where it's high. So really the direction of transport is important too. Passive is no energy required. Active does require energy. But in passive transport, you also need to know molecules move toward their lower concentration. Molecules move toward their lower concentration in passive, and then they move toward their higher concentration in active. All right, so in passive transport, again, no energy is required, and molecules are going to move toward their lower concentration. And we say that is with or down their concentration gradient. Concentration gradient is just a difference in concentration. So molecules are going to move down or with their concentration gradient when they move toward their lower concentration. So passive transport includes simple diffusion, facilitated diffusion, or sometimes called facilitated transport, and osmosis. All right, so with simple diffusion, molecules are going to move through the phospholipid bilayer. When molecules move, I was seeing if, here we go. All right, this shows simple diffusion. When molecules move from their high concentration to their low concentration, and they move directly through the phospholipids, we call that simple diffusion. And the only molecules that can do that would be small, and they would be lipid soluble. So they're going to be nonpolar molecules, nonpolar covalent molecules. So examples of molecules that move across the plasma membrane by simple diffusion are going to be, again, they're nonpolar, so they're lipid soluble, they're hydrophobic, and they're going to include gases, oxygen and carbon dioxide gas, and they're also going to include lipids like steroid hormones and cholesterol and fatty acids. Sometimes if the substance is small enough, like water can pass through the phospholipid bilayer even though it's polar because it is small. So sometimes molecules are small enough they can pass through the phospholipid bilayer anyway, even if they are polar. Okay, we don't need that page. I do want to go over the homeostatic imbalance. It says if the plasma membrane is damaged, substances can diffuse freely into and out of the cell, and that compromises concentration gradients. In other words, concentration gradients need to be set for certain substances because it's necessary for things like muscle contractions and nerve impulses. So when the concentration gradients aren't at the levels that they're supposed to be, then it can cause all kinds of different things to go wrong. So it says burn patients lose precious fluids, proteins, and ions that weep or leak out from damaged cells. All right, so there's an animation on simple diffusion, which you can watch if you would like. It is slide 43. And then we're going to talk about facilitated diffusion. So molecules that can't move through the phospholipid bilayer, they can still move into or out of the cell with diffusion, but they have to have a carrier protein. So we call it facilitated diffusion because a carrier protein is helping out. So facilitated is transport across the plasma membrane with the assistance of a carrier or channel protein. And it is passive, so no energy is required. If the substance moves through a channel protein, we call it channel-mediated facilitated diffusion. There are certain channels for water called aquaporins. So water can move through a channel protein by facilitated diffusion. A lot of substances move through a carrier, so we call it carrier-mediated facilitated diffusion. You don't have to learn that whole term. You can just call it facilitated diffusion because it's almost always a carrier protein instead of a channel protein. But basically, the substances that move through the plasma membrane by facilitated diffusion would be glucose, which is polar, and amino acids and ions, which are all glucose and amino acids are polar molecules. Ions have a charge, so any of those can move through the phospholipid bilayer. And I see a mistake. So I am going to mark through this, and I'm just going to tell you what it's supposed to be. Okay? So you can fix that word if you want to. I kept looking at it when I was reading the notes and trying to talk about the notes, and I was like, it was confusing me. because glucose, amino acids, and ions are not hydrophobic. They are hydrophilic. They're water-loving. So hydrophobic is nonpolar molecules, which are lipid-soluble. So this word should be hydrophilic. Glucose and amino acids are polar molecules, and ions have a charge. So they are hydrophilic, and they're able to move through a carrier protein because they can't actually move through the phospholipid bilayer. All right, so the only thing that I will say is there is a note about the different type of channel proteins. Some are leakage channels, and that means they're always open, and molecules just leak either into or out of the cell. And the channel proteins can also have a gate that is either controlled by a chemical binding to it or by a voltage change. So a voltage change can open the gate. So the channel proteins in facilitated diffusion can be leak channels or gated channels. And what we're looking at here are carrier proteins, and this is glucose. Glucose is a hydrophilic substance, which is soluble in water, so it can't move through the phospholipids, but it moves from its high concentration outside the cell to its low concentration inside the cell by facilitated diffusion with a carrier protein. And so the carrier will bind to the glucose, and once the glucose binds chemically with that carrier, the carrier will change shape, open up, and release the glucose inside the cell. And a channel is just a pore or a hole or a channel through the protein. And we're looking at some small molecules which are moving through. These could be molecules of water, but they're moving through the channel with their concentration gradient from high to low. Osmosis is specifically the diffusion of water, and water is going to move through the plasma membrane through specific water channels called aquaporins. There are leak channels that are always open, and water is the only substance that can move through them. But sometimes, in addition to aquaporins, water can actually diffuse through the lipid bilayer because it is so small. So osmosis is the movement of water that occurs due to differences in concentration of certain solutes on each side, on the outside the cell and inside the cell. So this is just a picture of aquaporins. But water is going to follow the rules of diffusion or passive transport. The water itself is going to move from where the water concentration or the amount of water is high to where it's low. But it's really easier and makes a little bit more sense to talk about water moving toward areas of high solute. So if you can remember this, water follows solute. Water is going to move toward the area where the concentration of solute is highest and the concentration of water is lowest. So water does move toward its low concentration, but sometimes it makes a little more sense, especially when we get into some of the body systems like the urinary system, to remember that water is going to follow solute. It's going to move to the area where there is the highest concentration of solute. Okay, so that can help us understand what will happen when we have solutions that have two different concentrations. So what we're looking at is a U-tube. This is an actual lab glassware that's used in lab to show osmosis. It can be used to show osmosis. So I'm going to blow the picture up so we can see it a little bit better. In this U-tube, we have a concentration of some solution. I don't know what this solute is, or it tells us. I'm sorry. The red solute molecules are sugar. So we have a sugar solution on the left side that has where the sugar has a lower concentration, and then we have a higher concentration of sugar solution on the right side. Then we have a membrane separating the solutions that the sugar can't cross, but water can. So if the membrane is permeable to sugar, sugar will move across that membrane until it's evenly distributed. But that's not what I wanted to show you. I should have looked more closely. What I really want to show you is a picture where the holes in the membrane are small, so the sugar is not able to move from one side to the other. So when the solute molecules can't move through the membrane, then water will. And water is going to follow solute. Remember that, water follows solute. Water is going to move to the area of higher solute concentration, and we see that happen here. So the level of water on the left side will go down, and it will rise on the right side, and it will basically move to the right until the solutions are in equilibrium, until they're basically the same concentration. So water follows solute. Water moves toward the area of higher solute concentration. And the next thing we're going to do is talk about tonicity. Tonicity is the ability of, like when cells are placed in certain concentrations of solutions, it's the ability of a solution to cause that cell to take on water or to release water. So it causes the cell to swell or shrink. So tonicity is related to the concentration of solution inside and out of the cell. If the solution that the cell is placed in is isotonic, then it has the same concentration as the cell, so the cell stays the same size. If the solution is hypertonic, it has a higher solute concentration than inside the cell. So remember, if the solute concentration is higher on the outside of the cell, water is going to move out, and it's going to cause the cell to shrink because it loses water. In a blood cell, in a red blood cell, this is called crenation, when the cell shrinks because it has been placed in a hypertonic solution. And then if you place a red blood cell in a hypotonic solution, that means the solution has a lower solute concentration than inside the cell. So an example of a hypotonic solution would be distilled water, because it would have 0% solute. So a hypotonic solution has a low solute concentration. Since water follows solute, the higher solute concentration is inside the cell. So water is going to move into the cell following the higher solute concentration, and it can cause the cell to burst, and we call that lysing. In a red blood cell, we call it cytolysis. And here's a picture that shows you a red blood cell placed in an isotonic solution, stays the same size. Red blood cell in a hypertonic solution undergoes crenation and shrinks. Red blood cell in a hypotonic solution swells up, and it can burst or lyse. And the animation on osmosis, I highly recommend you watch this animation. It is on slide 55, and this is where I'm going to stop this recording, and we'll start the next recording with slide 56.